Innovation is the origin of infinite possibilities. Ideas evolve into inventions. Inventions bring about revolutions. Revolutions transform lives. Since centuries, the vulnerable human race has been fighting numerous diseases, shielding itself with the armor of healthcare innovations. Vaccination was invented as a method of fighting infections with their own weapons. It gave us an impenetrable shield, immunity, and we realized true health isn't the management of diseases, it's the ability to prevent them. Over the years, vaccination protected mankind, but is still unable to match the pace of rapidly spreading diseases. At the cusp of the new millennium, a need was arisen to explore the next level in the science of vaccination. Cadila Pharmaceuticals rose to the occasion and joined hands with a leading US-based vaccine company, Novavax, to revolutionize vaccination. Thus, CPL Biologicals was born. Mr. I.A. Modi, the legendary medicine man of India and founder-chairman of Cadila Pharmaceuticals Ltd, pioneered this initiative to provide high-quality affordable vaccines to people through world-class research and innovative manufacturing.
Cadila, in all respects, has come in the top gear. We have made a history in raising vaccine facilities for production in just 1 year of the international standard. Easiest, best and most economical and most effective, we will be able to give the products to the world that we all succeed in our efforts.
CPL Biologicals has constructed a state-of-the-art vaccine facility at Dholka, Gujarat, that currently has the capability of producing 25 million doses of novel vaccines every year using virus-like particles, VLPs, and protein nanoparticle-based technologies. Radicalizing vaccination. VLPs and protein nanoparticles are warriors of the vaccine revolution, spearheaded by CPL Biologicals. They are far superior to the traditional vaccines. They mimic virus or infectious agent as a structure and closely match an individual viral strain or protein antigen. CPL Biologicals vaccines can trigger a more robust immune response. Zero scope for infection. A disease-causing virus is not needed to produce a VLP or nanoparticle vaccine. Only the genetic sequence of the virus is used to quickly create the vaccine against it. Since these vaccines do not contain virus nucleic acids, they cannot replicate and therefore they present no threat of infection to a person being vaccinated. Influenza vaccine, the need of the hour. Looking at the global spread of influenza recently in the form of pandemics like H1N1, swine flu, H7N9, avian flu, H5N1, bird flu, and seasonal influenza, a threat to vulnerable population. CPL Biologicals' primary focus is on developing the influenza vaccine candidates in India using VLPs. VLP-based vaccinations, the superior science. Conventional influenza vaccine manufacturing method requires growing an actual influenza virus, acquiring embryonated chicken eggs and adapting the virus. This old method requires up to 8 months lead time to produce a new strain of virus. With new strains of influenza virus emerging every year, it is crucial to develop faster methods of vaccine creation. VLPs ensure a recombinant, fast, simple, and efficient manufacturing process. While traditional methods pose a difficulty in matching the circulating influenza strain each year, VLP-based method ensures exact match to the wild circulating virus and protection against drift. VLP-based vaccines have a broader immunological profile as compared to traditional vaccines, whose efficacy is questionable in certain age groups. Potential immunological advantages of influenza VLPs. VLPs contain three immunologically important proteins. HA, neutralizing antibody which prevents infection. NA, antibody response which reduces disease severity. M1, cell-mediated response which reduces disease severity. Current flu vaccines provide response almost entirely against a single flu protein, HA. VLP's small particle size permits processing by immune system in multiple ways. Dendritic cell processing and direct stimulation of lymphocytes in lymph nodes. Current flu vaccines are made of soluble proteins and not processed to create cellular immunity. Genesis of the vaccine revolution. The groundbreaking influenza vaccine marks the genesis of the vaccine revolution. The ensemble of highly experienced scientists and industry veterans at CPL Biologicals are striving hard to develop VLP and nanoparticle-based vaccines against diseases such as rabies, varicella zoster virus, hepatitis A and E, which are endemic to India. With the dawn of groundbreaking discoveries such as the use of VLPs and nanoparticles in vaccination, a health utopia isn't a far-fetched dream. A disease-free life isn't just wishful thinking. Revolutionizing healthcare isn't a long-term vision. The future of healthcare is in the making. CPL Biological Private Limited. Novel, affordable solutions for global healthcare.